For the urinary anatomy and physiology lab, you're going to read both these chapters, and these are the things you need to know. So you must be able to identify on a model some of these things, and then histologically you'd look at the kidney, cortex, ureter, and urinary bladder. And then for physiology, you will read up on these things, and you will be doing an activity on a urine analysis. So let me first kind of go over the anatomy part of it. And so let's take a look here. So here when we are looking at the kidneys, so we look at the kidneys up here, they have an outer capsule which is present here. This is the medial border, so this area is the hilum, which you can see, which has the renal artery, the renal vein, and the ureter coming out. This thin strip on the outside is the cortex. Then we have the pyramids, which are these triangular structures with the base. This is the base of the pyramid towards the cortex. And the apex are these little pointed areas up here, which is called the papilla of the pyramid. So this part here, this is the papilla up here. This is the papilla of the pyramid. And then a part of the cortex comes down in between these pyramids. So these areas where the cortex comes down is known as the renal column. So this part here is the renal column, for example. So here where these blood vessels are seen, this is the renal column, okay? A lobe of the kidney is the pyramid with the cortex and, a bit of the renal column on either side up there. Now here, when you look at the blood supply of the kidney, so we have the renal artery, which gives off segmental arteries, which then become lobar, and then they go on to going in between. So they're called interlobar arteries. And these branch out, and these branches meet here where it's called the arcuate artery. And the arcuate artery, as you can see, is between the cortex and the medulla. And then this arcuate artery gives off single branches, which are known as cortical radiate arteries, or also called interlobular arteries. Coming off from this interlobular arteries are the afferent arterioles, which go to the glomerulus. And if you look up here, the glomeruli, as you can see, are all in the cortex. And so is the proximal so let's follow this glomerulus in the cortex, proximal convoluted tubule in the cortex, the loop of Henle comes down into the medulla, distal convoluted tubule in the cortex, and then the distal convoluted tubule drains into the collecting duct. Okay, so therefore in the cortex you will see glomeruli, you will see the DCT, the PCT, and the collecting duct. In the medulla, you will see the collecting duct and the loops of Henle. So when you look at the histology, that's what you're going to be looking at. Then let's come down. So again, same thing up here, showing it really well. So here you can see the papilla. And what you also see is then as this nephron comes down and urine, the filtrate passes through the collecting duct and it comes down here. By the time it reaches the collecting duct, and comes to the bottom, we call it urine. It drains into these little tubes, and the tubes are shown better here. So we begin with minor calyces. Each minor calyx, as you can see, is cupping a papilla. So the collecting duct will pass through, and there are holes in the minor calyx through which the urine from the collecting duct passes into the minor calyx. A few minor calyces join to form a major calyx. So here's a major calyx, here's another one, here's another one. So you can see these two are forming this major calyx. And then the major calyces lead to the pelvis of the ureter, which is this funnel-shaped structure which comes down and becomes the ureter proper. So you can see the same thing here. So minor, major calyx, and pelvis. Now once the ureters come down, they pass into let's go down here. So ureters come down and they pass into the bladder. And in the bladder, when you look at the wall of the bladder, you can see that it is thrown into folds. This is the muscle of the bladder, which has a special name called the detrusor muscle. And in the bladder, we have a smooth triangular area on the posterior wall, which is called the trigone, which has the two ureteric orifices up here near the base, and at the apex is the internal urinary or urethral orifice from where it leads out into the urethra. Okay, so that is the whole tract. Now here are again some models on looking at some special features. So up here you can see the same thing. So this is the arcuate artery giving rise to the interlobular or cortical radiate, afferent arteriole. So here is the glomerulus. Always identify the glomerulus and follow that along. So here is the proximal convoluted tubule, then that will become the loop of Henle and will go around. Same thing we can see here. So here, if we take a look, so here is the arcuate artery, interlobular, afferent arteriole, glomerulus. This would be the efferent arteriole. So let's follow one of them. So here is the glomerulus. So these round structures are supposed to represent the glomerulus. So see here the glomerulus. Here is the afferent arteriole. That would be the efferent. From the glomerulus, this is going to be, this orange one is going to be the proximal convoluted tubule. And then that comes down as the loop of Henle, goes back up. So this is the descending, ascending limb of the loop of Henle. This becomes the distal convoluted tubule, which drains into the collecting duct. So you always see the distal draining into the collecting duct. So up here as well, so this distal draining into the collecting duct, okay? Here, the efferent arteriole is breaking up into this capillary plexus called the peritubular plexus, which is around the tubules. And then when you come down here, these capillaries become straight and we call them vasa recta. So again, here you can see glomerulus, proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct. So that is on that part. Now here, when we look at the glomerulus and the Bowman's capsule, which together are known as the renal corpuscle. So here is the afferent arteriole, which is a wider arteriole, efferent narrower, so that there's a pressure buildup here in the glomerulus itself, so that filtration can occur. And in this glomerulus, you have special cells which are known as, so in this afferent arteriole, those special cells are called JG cells, which monitor blood pressure because blood flows through it. So it can monitor blood pressure. And the distal convoluted tubule, as it comes, actually, it's usually not shown very well, but maybe let's see whether it shows up here. So the distal convoluted tubule actually comes very close to the glomerulus. So those cells of the distal convoluted tubule become taller, and those are known as macula densa cells. And since filtrate passes through the distal convoluted tubule, what this macula densa cells monitor is the solute content of the filtrate. So that's important to know. And here, when we look at the Bowman's capsule, so you can see it's cup shaped. So here's the cup, the outer layer of the cup, which is the parietal layer, and this is the inner layer of the cup, which will be the visceral layer. But the visceral layer will go over each of these capillaries like this. So you can see it goes over each of these capillaries, like that. And it's got a name. They are called podocytes of the visceral layer. So as blood flows through, it has to pass through the endothelium of the capillary and the basement membrane, and then the basement membrane and the visceral layer of this Bowman's capsule. So that's what makes the filtration membrane. And it passes into this space, which is called the capsular space or Bowman's space, or also called the glomerular space. Okay. So now that you understand that the glomeruli are all in the cortex, let's look at the histology. So here also you can take a look at the whole tract, and let's take a look at the histology. So when we look at the kidney, in the cortex, you will see glomeruli, which are these flower-shaped structures, which you can see here, and surrounded by that capsular space, and this outer layer is the Bowman space. So all of these flower-shaped structures are glomeruli, and the proximal convoluted tubules have very tall cells, distal have narrower cells. So you can see the lumen in distal convoluted tubules, whereas in proximal convoluted tubules, all of these where you can barely make out the proximal convoluted tubule lumen, those are the proximal convoluted tubules. Okay, so where you can see the lumen, that is a distal convoluted tubule, And typically collecting ducts also have a lumen, so you could call it collecting or distal, but collecting ducts are usually cut longitudinally. So you see, when you see a long structure with a lumen, that would be the collecting duct. Okay. In the medulla of the kidney, you only see the loops of Henle and the collecting duct. So notice all these collecting ducts with well-defined lumen, and you really can't in this picture make out the loops of Henle. If you manage to see something in the lab, you would have something with a very thin lining, that would be the thin segment. If it is a thick lining, that would be the thick segment. But you know that this is the medulla because it does not have any glomeruli. If you don't see glomeruli, you can safely call it the medulla. When we take a look at the ureter, the bladder is the same, but I use the ureter as an example. The epithelium is transitional, so it is thick epithelium, and the outer or the topmost layer of the epithelium has a little cuticle on top, which is impervious or waterproof, so it doesn't allow water in the urine to pass through and get into the walls. Also, this epithelium, the transitional epithelium, is capable of stretching, which is helpful in the case of the ureter and the urinary bladder because they hold urine. So that's important. They have a very, very thick layer of smooth muscle. In the ureter, it's just smooth muscle, doesn't have a name. In the bladder, remember, it's called the detrusor muscle. In the bladder, you would not be able to see the entire lumen. So all you would see is this, just a small part. And then this outer layer is called the adventitia, okay, which is just connective tissue. So that is the histology. So those are some of the things that you have to pay attention to. So read all of this. And then when you come to urinary physiology, you should know filtration, which occurs, as we said, through the filtration membrane into the capsular space. So it occurs into this capsular space. As it passes down, things get filtered, but they need to go back into the system. So from here, what happens is they go through the renal tubule into the interstitial tissue and back into the blood vessel. So we call it reabsorption because absorption already occurred in the digestive tract. So reabsorption is a process going from the lumen of the tubule into the blood vessel. Sometimes what happens, some substances pass out, but they need to be removed. So they couldn't get filtered. So as the blood is traveling through, it gets secreted into the tubule lumen. So that's called renal secretion, which is in the opposite direction. So from the blood vessel into the lumen and out in the urine. So that is secretion. So you should know the direction of flow of the filtrate in all of these. And then you also will do a simple urine analysis, like what does normal urine look like? What does someone with glucose in the urine show up with, and protein, which are not normal. You normally do not see protein and glucose in the urine. And then we also need to look at ADH and aldosterone importance of them in urine formation. So that you will be looking at up here so this is the video on the histology, and you will look at how urine is formed. So this gives a good idea of reabsorption and filtration. And here we are looking at the action of aldosterone and ADH. It talks mainly about ADH, but aldosterone acts a little bit similar. So it basically reabsorbs sodium, but water moves with it. So, again, urine becomes concentrated. So watch this video, and then you're going to do this. I mean, you don't, when you do it, you won't resume. I had done it, so that's why it's saying resume. But you will kind of do this activity, and then there'll be little questions that you're going to answer at the end of this. Okay, so that's what you're going to do for this urinary lab, and any questions you have, feel free to ask me.